Have you ever wished you could find a single faulty gene inside our bodies and just flip an off switch? Well, today we're going to talk about how to do pretty much exactly that. We're hacking our own genetics with a really powerful tool our cells already have. And no, this isn't science fiction. It's a fundamental biological process called RNA interference. Let's get into it.
Really think about that for a second. The power to pinpoint one specific gene that's causing all the trouble, for a rare genetic disorder, a persistent virus, maybe even a gene that's driving cancer, and just silence it. This very question is what's fueling a massive revolution in medicine. And the answer, it turns out, was inside our own cells all along.
That's right. Our cells are already equipped with this incredible machinery. It's a natural, incredibly precise, and super efficient system for defense and regulation. We call it RNA interference, or RNAi for short. And once you understand how it works, a whole new universe of medical treatments opens up.
Okay, so let's start with the basics of this amazing cellular tool. What exactly is this off switch? So what is RNA, really? It's a natural process where the cell uses tiny little RNA molecules to shut down a specific gene. Now, the key here is how it does it. It's not messing with your DNA. Instead, it intercepts and destroys the message that comes from the DNA. Think of it like intercepting a memo before it reaches the factory floor. It's an incredibly specific way to silence a gene.
And the speed at which this technology developed is just mind-blowing. It was first discovered way back in 1998 in these tiny little worms by Andrew Fire and Craig Mello. Just 8 years later, boom, they won a Nobel Prize for it. And then, only 12 years after that, in 2018, the very first drug based on this tech gets approved. That is a lightning-fast journey from a discovery in a worm to a drug that changes human lives.
To really get how this works, we need to meet the two main players in this little molecular drama. I like to think of them as the blueprint and the blueprint shredder. So on one side, you've got messenger RNA, or mRNA. This is your blueprint. It's a temporary copy of a gene's instructions that travels from the DNA out to the cell's protein-making factories. Its entire goal is to be read so a protein can be built. But on the other side, you've got our blueprint shredder, small interfering RNA, or srRNA. This little guy is a tiny, double-stranded piece of RNA that has one job and one job only: to act as a guide, find a specific mRNA blueprint that it matches, and make sure that blueprint gets torn to shreds. It's basically a precision-guided missile for genetic messages.
All right, so how does the cell actually use this blueprint shredder to silence a gene? Let's walk through the whole process step by step. It's actually a pretty elegant system.
Okay, so it all kicks off when a long, double-stranded RNA molecule shows up in the cell. The cell sees this and gets suspicious, because that's often a sign of a virus. An enzyme called dicer, a great name, right, acts like a pair of molecular scissors, chopping that long RNA into small pieces. These are siRNAs. Then, one of those little pieces, the guide strand, gets loaded into this incredible protein machine called the RISC complex. Now RISC is armed and dangerous. It patrols the cell, constantly scanning all the mRNA blueprints. When it finds an mRNA with a sequence that is a perfect match to its guide, bam, it slices the mRNA, completely destroying it. The blueprint is gone, the protein can't be made, and the gene has been effectively silenced.
And this quote just nails it, doesn't it? The RISC complex isn't just a shredder, it's a programmable one. That's the beautiful part. By simply feeding it a different siRNA guide strand, you can redirect this incredibly powerful search and destroy system to target pretty much any gene you want. And that right there, that programmability, is what makes RNAi such a revolutionary tool for scientists and doctors.
Which, of course, brings us to modern medicine. How do we actually take this natural process that's happening in our cells and turn it into a life-saving drug?
Well, for years, this was the big problem. See, these synthetic siRNA molecules, our blueprint shredders, are super fragile. If you just injected one into the bloodstream, it would get chewed up and destroyed by enzymes in seconds. It wouldn't last. And even if it did survive, how would you get it into the specific cells you're trying to target? This delivery problem was the #1 hurdle.
The solution was frankly brilliant. It's something called lipid nanoparticles, or LNPs. Just think of them as tiny, fatty bubbles that act as a delivery vehicle. You tuck the fragile siRNA drug safely inside this protective sphere. The bubble shields it from all those destructive enzymes and lets it travel safely through the bloodstream to its target. And if this technology sounds a little familiar, it should. It's the exact same delivery system that was absolutely critical for the mRNA COVID-19 vaccines.
So, with this amazing delivery technology finally figured out, scientists could at last turn the incredible promise of RNA into a real-world medicine that helps people.
And that brings us to patisiran, the very first siRNA drug ever to be approved. It's designed to target a gene called TTR. In some people, a mutation in this gene causes it to produce a toxic, misfolded protein that builds up and causes a really devastating disease. So patisiran uses an LNP to deliver a custom-built siRNA to the liver, and that siRNA basically tells the liver cells, hey, stop making this faulty protein. It's a perfect example of using the body's own system to treat a disease right at its genetic source.
So if we can do this for one rare disease, what's next? Well, the potential here is just absolutely enormous.
I mean, just look at this list. This is just a small sample of the diseases where RNA therapies are being developed right now. We're talking about siRNAs designed to shut down genes that cause high cholesterol, genes that help cancer tumors grow, or even genes that viruses need to replicate. From very common problems like heart disease to a whole range of other rare genetic conditions, the ability to silence genes on command is creating entirely new ways to think about treatment.
And that really leaves us with this one final powerful question. We now have a technology that is for all intents and purposes a programmable drug platform. By designing a new siRNA, we can, in theory, target any gene that causes a human disease. So the question really isn't if we can do it anymore. The question is, what should we choose to cure next? What challenges will we take on with this incredible power to silence our own genes? Thanks so much for joining me.